Thanks, Justin, and welcome. And thanks so much to all of you for coming. So for many people who worked together in the lab, this is like getting the old band back together. So I think we're going to go on tour to other dance companies next. So as the sign indicates, all book sales proceeds go to the ODC. And I'd encourage you, this is the most recent new element in the ODC empire, but when you have time, you should visit the other buildings and the other functions. So several years ago with my colleagues Eric Stover, who is here, and Tom White, who couldn't be here, we did a book together called Silent Witness. And this book was highly technical, and it was oriented toward lawyers and judges and graduate students. But this book, Genetic Reconstruction of the Past, the new book, I wrote for a general audience. Now, I've been told that I didn't always succeed. There's some parts where the eyes tend to glaze over, but I think for the most part, it's accessible. And this book is also much more personal. And one of the things that I try to do in this book is impart the flavor of doing science, capturing the moment of discovery, that moment when you think this might just work. And I also try to discuss some of the properties of science as a human enterprise that distinguish it from other scholarly research. But the first thing I'd like to emphasize is that scientific research is a team sport, and actually many of the people who contributed and helped make the work that's in this book possible are here with us tonight. Estimates. Now, the other thing that I wanted to stress about the nature of the scientific enterprise is, I think, an underappreciated element in scientific research, and that is skepticism. We're all familiar with innovation and creativity and curiosity that drive scientific research, but skepticism is embedded in the scientific enterprise. And in experimental design, you always design what's called a control, which is essentially kind of institutionalized skepticism. It tells you whether the experiment you ran went the way you thought it did, and also whether the hypothesis you were testing is correct or not. So I'm going to actually show one example of a control that was critical in the development of PCR itself. Now, I know many of you are not scientists, so let me just very briefly explain what PCR, polymerase chain reaction, is. It's a technique for generating millions of copies of a specific sequence in a test tube. And it uses the basic elements that DNA replicates within the cell. It uses an enzyme known as a DNA polymerase that copies the DNA from a template. So that's a very brief introduction to PCR itself, but this book tells the story of how my colleagues and I at CETUS developed this PCR technology and how we applied it to forensics and evolutionary studies. Now, these two seemingly disparate areas rely on exactly the same kind of genetic data to reconstruct the recent past, the crime scene, or the ancient past. And you can see that in the table of contents. Part one is reconstructing the crime scene, and all those chapters have to do with the criminal justice system and the use of DNA. And I'm actually going to go over some of the specific cases that we worked on and what they really illustrate about the character of our criminal justice system. And part two then is about applying the same technology and the same kind of data to reconstructing the ancient past, how populations migrated, and how ultimately we now know modern humans emerged out of Africa about 75,000 years ago, spread to the Middle East, Europe, Asia, and ultimately Australia and the Americas. So the historian Jill Lepore, who some of you may know for her beautiful history of the United States called These Truths, wrote that history is the study of what remains, what is left behind. And well, DNA is what is left behind at the crime scene or in the fossilized bones of a Neanderthal skeleton, and the information embedded in the sequence of bases in the DNA can be used to reconstruct historical events. Now, some of you may be wondering why a genetics book is being discussed here at ODC. So I will proceed to tell you, this is from the preface, which discusses how I got interested in science and how I ended up at Cetus Corporation with all these characters in the audience. I had accepted the position at Cetus in large part because of Brenda. She had left a tenured position at Oberlin College to come west to live with me in San Francisco. She arrived with her 3 kids and 12 dancers, members of the Oberlin Dance Collective she had founded a few years earlier, in a big yellow school bus. So when my postdoc at Stanford was over, my priority was to find a job in the Bay Area. Cetus, which was a biotech company located in Emeryville, invited me to give a lecture and offered me a position. I anguished over whether I would be going over to the dark side by working for a for-profit company. But I was reassured by the fact that 2 of my Stanford mentors, Hugh McDevitt and Stan Cohen, were advisors, and most importantly, by spending time with many of the bright young Cetus scientists, notably Tom White, John Sninsky, and David Gelfand, who had recruited me. Their energy and enthusiasm were palpable, and the culture seemed decidedly more counter than corporate. So there I was in 1979 with my colleagues, we band of brothers and sisters, and we were trying to figure out what would be a useful and valuable kind of research project to work on. So what we did was create PCR. And as you know, PCR has become an essential component of basic research as well as medicine. You may be familiar with it when you were having your nose swabbed for a COVID test, and of course a lot of the early tests were infectious disease diagnostics. In those cases, we would amplify the DNA from a virus. In fact, my colleague John Sninsky led the team that developed the HIV viral load test, which was enormously important in addressing the AIDS crisis. And 2 other people who were here, Russ Higuchi helped develop a quantitative method of using PCR, and Randy Saiki developed a detection method of detecting specific targeted DNA. So it was really a team effort to develop the HIV viral load test. But what my lab was interested in was medical genetics. So we were looking at genetic variation in the human genome, not in viruses. I maybe should just make a little caveat that we did work on the human papillomavirus, the virus that causes cervical cancer, but virtually all my work was looking at genetic variation in human DNA. And among other, we were looking at genetic diseases like sickle cell anemia, where we were detecting specific mutations, but we were also looking at polymorphism in the human genome. What polymorphism is simply DNA sequence variation. And in terms of individual identity, for forensics or missing person analysis, if you want to see if you can match DNA from an evidence sample with a suspect, say, since well over 99% of the DNA in all of us is identical, you focus on those small regions of the human genome that are variable, that are polymorphic. And that that is how you can use DNA for individual identification. So having established a database of genetic variation and a technology that could distinguish DNA sequence variants, we were able to apply that to forensics. And as in so many cases in life, you could ask, how's a nice molecular biology or biologist or immunogeneticist like me doing forensics work? Well, it just happened that there was a forensic scientist, Ed Blake, who had a small lab in the same building that housed CETUS in Emeryville. And Ed and I would run into each other in the hallway, and he would tell me about his casework problems, and I would describe my basic research projects. And at one time he came to me and said, Henry, I've got these samples that I was sent from a case in Pennsylvania called Pennsylvania versus Pestinikas, which was a negligent homicide case where the owner of a nursing home, Walter Pestinikas, was accused of negligent homicide for one of the patients at this nursing home. And the prosecution had decided that there were two different autopsy samples, and in an attempt to cover up this negligent homicide, that the autopsy samples had been switched. So they asked Ed to see if he could see whether these two autopsy samples were from the same individual. Their hypothesis, I should remind you, was that they were not. So that turned out to be the first DNA case in the world, and it was simply because these samples were stored in formaldehyde. So all the tests that were available to Ed at that time were based on protein analysis. You could do ABO blood typing, you could do protein electrophoresis, but all the proteins in this sample had been denatured by the formaldehyde treatment. So I said, well, Ed, we could try DNA. We could try the current standard methods of trying to look at variation. But when we extracted DNA from these autopsy samples, they were all degraded. That is, all of the DNA fragments were very small. So I said, sorry, that's not going to work. And I then said we could try something that we'd just been working on. If I can find the right page. And this discussion is in the All Rise, DNA Enters the Courtroom chapter. So we were disappointed that the DNA was degraded and that you couldn't do any of the standard techniques. After my initial disappointment at this result, I suggested to Ed that we should try our newly developed method of targeted DNA amplification, PCR, or the polymerase chain reaction. My colleagues and I had just published the first account of PCR amplification of a specific human gene, beta globin, in a test tube using a DNA polymerase, an enzyme that copies DNA inside the cell, from the well-known laboratory bacterium E. coli a few months earlier. And that was paper Saiki et al. 1985, in which we amplified beta globin and carried out a prenatal diagnosis of sickle cell anemia. And now, 40 years later, Steve Mack and I are still doing prenatal diagnosis of sickle cell anemia and beta thalassemia, but this time we're looking, and this was work that I started with Sandy Calloway some time ago, looking at fetal DNA in the mother's blood, so a non-invasive prenatal test. But anyways, so I said, we got this method, we could try it. So after Randy Saiki, a young and very bright technician in my lab, had successfully amplified the beta globin gene, defining the narrow set of conditions under which Kary Mullis's idea might be made to work, we developed a system for amplifying and analyzing the DNA sequences of a gene involved in the immune system, the so-called HLA-DQA1 or DQ alpha locus. This is a gene that's involved in the immune system. The HLA genes are the most polymorphic genes in the human genome, and because, and we chose an HLA gene given my long-standing interest in immunogenetics. So because the HLA gene is polymorphic, we decided to see if this might be able to address the problem that Ed was having. So our first attempt, using the DNA polymerase from E. coli and the degraded DNA isolated from the formaldehyde-treated autopsy sample, failed. My technician at the time and for the following 33 years, Dori Bugawan, who is here tonight, then tried a newly isolated and more efficient DNA polymerase that my colleague David Gelfand had literally just purified from the bacterium Thermus aquaticus. This is the bacterium that lives in the Yellow Spring hot springs. And so when we tried this new enzyme, to our delight, we were able to amplify the polymorphic HLA-DQ alpha gene. So we were extremely excited, and as it turned out, the two autopsy samples matched. So the prosecution asked for the test, the result favored the defense. So there was actually no admissibility hearing. Admissibility hearings where there's a challenge to the introduction of a new technology to the jury. So there was no admissibility hearings, and the results were presented to the jury. Now, Walter Pesanikas was ultimately convicted of negligent homicide, but as it turned out, this was the very first introduction of DNA evidence into a courtroom. So because there was no admissibility hearing, I was very blissfully ignorant and naive about the nature of the admissibility hearing. And in all other cases that I worked in, I testified in what's called an admissibility hearing, where the judge gets to decide whether the evidence that's presented can be shown to the jury, whether it's reliable enough, whether there's a scientific consensus, and so forth. And my experience in these admissibility hearings was that it was an extremely contentious and adversarial system, where there would be an expert working for the prosecution and another scientific expert working for the defense. And if the evidence favored the defense, the prosecution would say it was totally garbage. And if it favored the prosecution, the defense would say it was unreliable. So that was my experience for the first few years. But before, and I'm going to discuss a few cases where I testified, but before that, I'm going to subject you to some science. And I know many of you are not scientists, so do not be intimidated by this. In the book, this is in the appendix, so you can safely ignore it. But you're here with me today, and I'm going to guide you through this. So it's not as bad as it looks, but this data actually illustrates 2 points that I want to make. It illustrates the role of the control in experimental design, and it shows how the introduction of this new thermostable DNA polymerase that I mentioned transformed PCR. So if you focus your attention just on the left part where it says Klenau, and where there's kind of a smear there, that's actually the technical word for this smear. You can see what our first attempts at doing PCR work. And in order to understand this, I have to explain to you one more element of PCR. So PCR is a series of cycles in which each cycle has 3 individual components. So the first thing you do is you have a double helix, 2 strands that are hydrogen bonded, and you can disrupt the hydrogen bonds and separate the 2 strands of DNA by just heating the reaction to say 97 degrees centigrade. So that's the first step. The second step, you add what are called, this is how you target the DNA that you're going to amplify. You add short pieces of synthetic DNA that are called primers, and they bind to the region that you're interested in. So in this experiment, they're binding to a small segment of the beta globin gene. And then you add a DNA polymerase, and you synthesize one strand that's complementary to this strand, and synthesize another strand that's complementary to that strand. And now you started out with one double helix, now you have two double helix. And then you just repeat and rinse. Basically, you repeat that cycle over and over again, and if you do 10 cycles, is an exponential reaction, hence the name chain reaction. You've created a million copies of this one DNA molecule. So that's the way PCR works. And when we were analyzing what we actually synthesized in a test tube, we would run it on a gel to separate the large DNA fragments from the very small DNA fragments. So this shows 20 cycles, 25, 30, 35 cycles of PCR on human genomic DNA, and this is our negative control, that if everything works well, there's nothing here. That's human DNA that's a homozygous deletion for beta globin, that there's no beta globin. So if you think you want to amplify beta globin, there should be nothing at all in that lane. So if you just looked at that, you decide, hey, this is all noise. This idea doesn't really work at all. But then this is the work of Randy Saiki. Randy and I decided, well, maybe there's a signal in the noise here somewhere. So we used a radioactive probe that is a short piece of DNA that's radioactively labeled that's specific for beta globin. And that's shown here. So you can see, oh, if you have a probe for beta globin, what you're seeing accumulate is in fact a discrete fragment of the predicted size. So by using this probe, we were able to basically discern a tiny signal in a vast amount of noise. So, you know, we went from 1 in 10 to the 7th to 1 in 100. So the beta globin was only 1/100th of what we amplified, but that was still a huge success, a huge enrichment over the amount of that DNA in the human genome, which, as I said, is about 1 in 10 million. So that shows you the value of a control. You're testing your hypothesis. You're asking, does the reaction really work the way we diagrammed it? And of course, it's not very specific because this is a huge smear. But you can see that if you look for beta globin, you actually were able to amplify it. And the other thing that this slide shows is the transformation in the PCR itself that was made possible by implementing the use of a thermostable DNA polymerase. So the DNA polymerase from Thermus aquaticus, which lives in the hot springs, is resistant to high temperature, and you can do the DNA synthesis reaction at 70 degrees instead of 37 with the E. coli polymerase. So you can see here that just introducing the Taq polymerase alone makes the reaction much more specific. We are now getting the predicted discrete band, but what's kind of interesting in our negative control, where there's no beta globin, you still synthesize some DNA. But so the important thing is Taq polymerase makes the reaction much more efficient and much more specific. And the reason it does that is because you can do the DNA synthesis mediated by the polymerase at a much higher temperature. So these short oligonucleotide primers bind more specifically. And I guess the other point that's made in this slide is it transformed PCR from a very clunky, labor-intensive reaction where we had to, because each cycle starts with heating the reaction at 97 degrees, so that of course inactivates the E. coli polymerase, and you'd have to open the tube and add enzyme. But with the Taq polymerase, it was thermostable. You could just add everything to the reaction tube and cycle the temperature. So the instruments that were developed to automate this reaction were called rather unimaginatively thermal cyclers. But the enzyme in these instruments transformed PCR into kind of, from an esoteric, complicated research technique to basically an essential tool of molecular biology. So thermostable DNA polymerase made such an impact, it was on the cover of Science magazine as Molecule of the Year. Of course, we were all hoping it would make the cover of People magazine as sexiest Molecule of the Year. So coming back to the issue of comparing the genetic profile of the evidence to a suspect, you know, there are two outcomes. It's either match or not. So those are sometimes called an exclusion, where they're different, or an inclusion, where they're indistinguishable, where they're the same, or where they're a match. And if it's an exclusion, that's the end of the story. No statistics required. That sample did not come from this dude. But if it is a match, if it's an inclusion, the significance of that match depends entirely on the frequency with which that particular genetic profile can be found in the general population. So to interpret forensic evidence, you need a lot of population genetics data and how that's distributed throughout different populations. So one of the points that I try to make in this book is the commonalities, the connections between forensics and evolution. They seem like very disparate fields, but as I said, they use the same kind of DNA technology. And they both are about reconstructing the past, the recent past or the ancient past. But so I'll just give you one example where I already mentioned why population genetics information is important to interpret a match. Steve Mack and I were looking at genetics, population genetics of populations in Asia and the Pacific, and we noticed that if you looked at Papua New Guinea and Australia, the genetic profile, the gene frequency distribution in Papua New Guinea highlands was much closer to the people, the Australian Aborigines, than it was to people in the Papua New Guinea lowlands. So that's the kind of genetic observation that is consistent with certain narratives. And the history that is illustrated by this genetic relationship is that in earlier times, Papua New Guinea and Australia were one single landmass, and it was initially populated by groups that settled in what are now the Papua New Guinea highlands. But later, when at the end of the Ice Age, when the waters rose and separated these two landmasses, Papua New Guinea and Australia, those people in Australia and the Papua New Guinea highlands shared their origins. But the lowlands of Papua New Guinea was populated much later by colonists from elsewhere. So it just shows how the same kind of data can be used to interpret a match or to make inferences about human migration and human history. So I had mentioned that in Pennsylvania versus Pestenicus, there was no admissibility hearing. So the first case that I was involved in was California versus Mello, a rape murder case, where the prosecution there were two suspects. They'd made a deal with one, and they were prosecuting the other for rape and murder. And if they could convict him of rape and murder, they would ask for the death penalty. But unfortunately for them, the HLA-DQ alpha genotype of the semen sample didn't match Mello, the suspect that they were prosecuting. So the prosecution argued that this is all garbage, junk science, and so forth. but fortunately the judge allowed it. So it was shown to the jury, and Mello was convicted of murder, but not of rape. But the irony in this case is that the DQ alpha genotype did match the guy they made the deal with. So that was kind of a deep irony. And there was another irony in the next case where I testified. This was a case where the DNA evidence matched the guy. It was California versus Mack. It was also a rape murder case. No relation. No relation. Anyway, so it matched the guy, and of course he said, "Oh, I never knew this woman. I'm innocent." And so in the course of the admissibility hearing, I had to present a lot of data, but the judge finally decided that because the FBI was not doing it yet, he was going to rule it inadmissible. So I hadn't managed to convince the judge, but apparently I did convince Mack, because he changed his testimony and said, yeah, he knew this woman, he had sex with her, but it was consensual and had nothing to do with the rape and murder. He was convicted of rape and murder. So as I mentioned earlier, that one of the things I noticed was really an adversarial system where you had dueling scientific experts. And I know in other countries, scientific experts evaluate new data and work for the judge, for the court, for the magistrate, rather than one of these advocates. In my experience, that would be a better system. And in these admissibility hearings, some of the rulings had the following property. This is the case, the People versus Castro, where it was a murder, there was blood found on Castro's watch that was not his, and it allegedly, according to the DNA evidence, matched the victims. So what the judge decided at this time, and I saw the data and it wasn't super clean, so I think the judge in this particular case may have made the right decision. He decided that the exclusion was admissible, but the inclusion was not. Another way of putting it was inclusions were excluded and exclusions were included. But anyway, the fact that the blood on his watch was not his, so you might well ask, well, where did it come from? And Castro was convicted of murder. But that was just in the early days of admissibility hearings. These hearings are extremely contentious. They got kind of personal sometimes. Judges were more comfortable with allowing exclusions and inclusions. So one of the things that Ed Blake and I realized as we were doing this, that we were able to actually analyze samples from old cases. And so we ended up doing the first DNA exoneration in the world, which was the Gary Dotson case. Gary Dotson was convicted of rape, and the alleged victim recanted her story. She said later, no, I made that up. I had sex with my boyfriend, and I just said this to cover it up. Nonetheless, the prosecution declined to overturn the conviction. And Alec Jeffreys, it's now Sir Alec Jeffreys, who is the pioneer of what's called DNA fingerprinting in the UK, analyzed this, but his technique didn't work on this DNA because once again, as in the Pesnikas case, the DNA was degraded. So we did the case and showed that it was an exclusion, and finally Gary Dotson was fully exonerated. But it took a long time, and as I said, one of the things I came away with was thinking that law enforcement does not like to say they were wrong. It's like a country song, it's my story and I'm sticking to it. They just, it takes a lot for them to overturn something. And I'll give you another another example, which is a very dramatic one. It has its own chapter, the Earl Washington Jr. case and the problem of mixtures. So it's such a dramatic and important case that I gave it its own chapter and also its own slide. Now, this is not the Earl Washington Jr. case. This is just an example of the HLA-DQ alpha test. You see there are blue dots, so it's basically a way of inferring sequence variation in the DNA that you've amplified. And I'm not going to go through it in any detail, just show you blue dots. And this particular case is an exclusion, where you can see that the pattern of the suspect does not match the pattern of the sperm fraction, and the genotype also doesn't match. So that's an exclusion, but you can also see that the epithelial, first of all, in a semen stain, you can separate the sperm fraction from the epithelial fraction. The epithelial fraction matches the victim, which is, of course, what you'd expect. So I am going to try to find the section that discusses the Earl Washington Jr. case. And while I'm doing that, this is a brief description of the case. Okay, so in 1982, Rebecca Williams, a 19-year-old girl, was raped and brutally stabbed to death in her apartment in Culpeper, Virginia. Before she died, she identified her attacker as a black man. A year later, Earl Washington Jr., an intellectually disabled 22-year-old African-American man, was arrested and charged with burglary and assault in a nearby county. The police accused Washington of Williams' rape and murder, and following interrogation, he confessed. At his trial, however, Washington maintained his innocence, but he was convicted and sentenced to death by a Virginia court in 1984. His execution was scheduled for January 1994. And his attorney, Barry Weinstein, who was convinced of his innocence, sent me the results and asked me to review the results of the semen stain and swab sample carried out by the Virginia State Lab. So that was the situation. And literally, he was scheduled to be executed a few days after I got the report. So, as you can see, I should say, the prosecution argued that his alleles are present in this. First of all, the profile of the sperm fraction is a mixture. You know that there's more than one person contributing to the mixture, because one person only has two alleles, two variants, one from his mother, one from his father. So if a sample has three alleles, you know it's a mixture. So the question is he present in the mixture or not? And the prosecution in the Virginia State Lab argued that his alleles were present because he's a 1.24, there's a 1.2 in there, there's also a 4. But the blue dots for 1.1 and 1.2 were much stronger, so they went together as a genotype. So the major genotype is a 1.1, 1.2, which excluded Earl Washington Jr. as contributing to this sample. So I sent in the report literally a day or two before he was scheduled to be executed, and Governor Douglas Wilder commuted his sentence to life in prison, but declined to pardon him. Then fast forward to 2000, there's now new methods called STR typing, short tandem repeats. They're just different ways of looking at genetic polymorphism, and many more than just a few genes we were looking at initially. And so that it also excluded Washington, but you could take those profiles and search a database, and they identify the guy, convicted rapist named Kenneth Tinsley. But it took until 2007, 23 years after Washington was exonerated, after he was convicted, when Tinsley confessed to rape and murder. So I learned three things from the Earl Washington Jr. case. One, that false confessions are not as rare as we have imagined. Prosecutors are very reluctant to overturn a conviction until the true perpetrator is identified, and mixtures are very difficult to interpret. I think that's all I'm going to say about part one in forensics. I'm going to talk very briefly about part two. And as I said, my interest I'd always been interested in forensics, but my involvement was made possible only because Ed Blake happened to have a lab in the same building. But similarly, because Ulf Ullenston, an evolutionary biologist from the Alan Wilson lab at Berkeley, came to my lab as a postdoc, and then later Steve Mack, who was also evolutionary biologist from the Wilson lab, came to my lab, I was able to explore some of the issues in evolutionary biology. And I'm just going to make a few comments here due to the time. But anyway, Alan Wilson was a professor at UC Berkeley, and he was really a pioneer of molecular evolution. He developed and applied the concept of a molecular clock, which is the attempt, if you compare sequences of the same gene from two different species, you can estimate the time at which they diverged. So that's really revolutionized evolutionary studies using DNA sequence information. But one of his most seminal observations was the out of Africa hypothesis. Some of you may be familiar with this, but this was looking at the diversity or the pattern of mitochondrial DNA diversity in human populations around the world. He and his colleagues inferred that the mitochondrial diversity you see in the rest of the world was only a subset of what you can see in Africa. And so the inference was that a small, well, maybe not small, but a population of individuals in Africa about 75,000 years ago, using molecular clock estimates, emerged from Africa to the Middle East. They populated the rest of the globe. And in doing so, as we now know, they interacted with Neanderthals, because Svante Pääbo, who was a postdoc at Wilson's lab at the time, and some of you may know he got the Nobel Prize for sequencing the Neanderthal genome. So that sequence analysis identified that it was a distinct subspecies of archaic humans, but it also identified that humans intermarry. I don't know about marriage, but they intermarry. I wasn't there to perform any of the ceremony, and I've done over 20. So there is Neanderthal genome in all of our genomes, about 2 or 3%. And one of the really amazing observations that the Pääbo group did is identify a totally new subspecies of archaic humans in a cave in Denisova, Siberia. This is based on a single finger bone, small finger bone. First they did mitochondrial DNA, which is much easier because every cell has thousands of copies and just 2 nuclear copies. But anyway, they showed that this was a species distinct from Neanderthal and distinct from modern humans, Homo sapiens. And one of their findings in this cave was really fascinating. It was a young woman, skeleton of a young woman whose mitochondrial DNA was Neanderthal, but half of her nuclear genes were Denisovan, half were Neanderthal. So basically, she had a Neanderthal mother and a Denisovan dad. And actually, one of the things that's interesting, Alan Wilson, among many other things he did, when he learned about PCR, he realized this was an incredible way to look at ancient DNA. And Russ Seguchi, who's here, did the first ancient DNA, a quagga, a museum skin from an extinct species similar to the zebra. But Russ and Svante Paabo and Alan wrote a review about PCR and molecular evolution, and they used the phrase, PCR made it possible to analyze ancient DNA and catch evolution red-handed. So, due to time constraints, I think I'm not going to, you can see, well, you can see some of the other chapters there. But I think what I'm now going to do is ask some of my colleagues to just say a few words. You know, they'll sit down and then we'll open up to questions from the audience. So I think I'm going to ask my friend John Sninsky, whom I've worked with. We were postdocs together at Stanford, and John is also, as I mentioned, the developer of the HIV viral load test. But John, if you want to say a few words.
Thanks, Henry. Very clear. Thank you very much. So I'm going to say a few words from Tom White, who regrets that he wasn't able to make it, but he was nice enough to provide a script for me to read. Tom greatly regrets that he can't be here this evening due to ongoing major road and bridge repair projects near his home in Rio Vista. He asked me to read his remarks for the occasion. Tom also wants to thank Henry for his epigenetic constructions in the past by officiating at his weddings to the late Leslie Scalapino in 1988 and to his current wife, Tina Arnold, in 2020. The adjective current wife, Tina Arnold, serves from Tom's recollection of Henry's alarm during an ODC fundraiser at the San Francisco Museum of Modern Art in 2002, when Brenda referred to him as her current husband. Tom's first wedding ceremony occurred in Henry and Brenda's backyard, and the second marital event in Kyla and my front yard. As Henry already mentioned, they met around 1980 when David Gelfand and the director of molecular biology research at CETUS Corporation asked Tom to help recruit Henry to join the company. Similar to what had brought Brenda Way and the Oberlin Dance Collective to the Bay Area, it was love for Leslie that had turned Tom to Berkeley in the middle of a 3-year postdoc at the University of Wisconsin in Madison, which he felt was not the Berkeley of the Midwest, but in fact, the Fresno. Over the following 3 decades, the four of them became wonderful and good friends, both as scientific colleagues and as Brenda and Leslie collaborated on several dance and poetry performances. Henry came to CETUS from Hugh McDevitt's lab at Stanford, as Henry mentioned, a mecca for research on the human immune system. The major focus of Henry's work was to study ways to genetically match donors and recipients of bone marrow transplants. In 1984, a creative but cantankerous scientist named Kerry Mullis conceived of a method he called PCR, again, as Henry mentioned, the polymerase chain reaction, to exponentially amplify short segments of DNA. Henry, Tom, and Norm Arnheim, the then head of the genetics department, were all doubtful that the method could be made to work. Again, as Henry mentioned, skepticism, but agreed that if so, it would have profound consequences as a research tool for diagnostics. Mullis was a poor experimentalist, so Tom and Henry assigned a research assistant, Steve Scharf, to help him. Through the efforts of Steve and then Randy Saiki, working under the scientific direction of Henry, PCR was proven to work in 1985 and was published in Science, a prominent scientific journal, in December of that year. For the next few years, improvements and new applications for PCR exploded, and as described in the genetic reconstruction of the past, these included the invention of PCR tests for forensics investigations and also for the study of human genetic variation or polymorphism among extant humans and our extinct human ancestors, such as the Neanderthals. Tom wanted to add a few comments about the book. Beginning in 2017, Henry asked Tom to read the early drafts of his memoirs chapters. It was a pleasure for Tom to provide comments and edits, such as, delete the phrase, "in my view," everywhere. It's your memoir. Everything is in your view. Another of Tom's suggestions was to be less nice to some of the characters who had acted unethically or intentionally perjured themselves in court or in the court of public opinion. Namely, to correct the record when those individuals wouldn't retract their lies or admit their misconduct. During this process, a second, more technical and specialized book, Silent Witness, Forensic DNA Analysis in Criminal Investigations and Humanitarian Disasters, was conceived, assembled, and co-edited by Henry, Eric Stover, and Tom. It was published by Oxford University Press in 2020. Unlike most scientists' memoirs, Henry's book is neither a self-hagiography nor unencumbered by excessive jargon. As a personal account of an outstanding scientist's career, it breaks the common cinematic and science fiction memes that we are all nerds, or quietly malevolent, or don't care about subjects outside of our research, like social justice or human rights. It's a stretch, but in its depth of description of scientists' lives, perhaps Henry's genetic reconstruction of the past is a contemporary reconstruction of Vasari's lives of artists. Thank you, Henry.
Thanks a lot, John. So I'd like to call on my friend and colleague Steve Mack. We've worked we've worked together for what, over 30 years, I think.
I will be brief. So I've been working with Henry for 32 years. And in reading the book, I realized that Henry's influence on my life has preceded the 32 years. I was in high school when PCR was invented, and I was a molecular biology student in high school, and I remember being very excited about PCR. And then I was in college when mitochondrial DNA, mitochondrial Eve was released, and I remember being so excited about PCR and molecular evolution that I wanted to come to California, to go to Berkeley to work with Alan Wilson, which guess what I did. Cut to the chase. And it's Henry is behind that whole thing. So you've been pulling me here to work with you. And so I just want to say a few words about the book. Read the footnotes. All right, the footnotes reveal Henry's inner self. So if you're wondering how Hieronymus Bosch relates to the genetic reconstruction of the past, or if you're wondering how, if you know what Occam's razor is, but you want to know what Occam's broom is, read the footnotes. And I just want to say thank you, Henry, for working with me. It's been a real pleasure, and this book is very exciting. So.
Well, thanks, Steve. So I'm very lucky. I'm still working with Steve now, so we see each other almost every day.
Yeah, our offices are right next to each other.
So the final speaker is my colleague, Eric Stover, who's the founder and the co-faculty director of the Human Rights Center at Berkeley. And Tom White and I sit on the advisory board, and it's been a great pleasure to work with Eric over the years. I just gave a lecture yesterday in his class.
Again, I'll work on keeping my comments brief as well. One of the things, many things that I admire about Henry is he doesn't suffer fools, so I'll be watching him here. Justin, you never had that problem? Okay, just checking. Okay, what I want to say is just a little bit, very as briefly as I can here, is to say that I'm not a forensic scientist. I have essentially, as I tell my students, a bachelor's degree in English literature and a PhD in misspent youth. But what I did end up doing early in the 1980s was working with the American Association for the Advancement of Science, setting up their science and human rights program. And this was a time when we could bring science into human rights. And I took the first forensic team down to Argentina after the military to begin the exhumation of the disappeared. And the reason I raise this is, which relates to Henry, is the fact that I had forensic anthropologists, radiologists, odontologists, and so on. And some of you will remember, of course, back then it was the bone versus molecule debate. Was these forensic scientists, they were looking at skeletal remains to do identification, determine manner and cause of death. But what came up in the late 1980s, in fact, I took a femur cross-section from a skeleton to a lab outside of Washington, D.C., and they couldn't extract DNA from it. But what became important was that the entrance of DNA into this work was so fundamental. And I was actually back in those years that I remember reading and learning about Henry's work, Tom White's work, and others here, because when we went on to Bosnia and worked in Rwanda, but in Bosnia, the International Commission on Missing Persons was established, and it has now identified nearly 67% of the disappeared based on DNA analysis.
Wow.
So I'm just trying to say how fundamentally important this work is. And I do just want to end by saying, to have Henry come into my class, it's a war crimes and human rights investigations class. We teach them how to think about multiple working hypotheses. No offense to the lawyers in the room, but if you're going on to a forensic, this could potentially be a mass grave. You work with multiple working hypotheses. You don't go in looking for any particular evidence. We ask the prosecution from the courts to go have lunch when we begin our work. But just to emphasize that during that process of seeing DNA being introduced, the contribution is often not appreciated around the world. And it really has been based on Henry's work and other colleagues here. So thank you.
Thanks a lot, Eric. So I'd like to open it up to questions for a few minutes, and then I'm going to conclude with a reading about Alan Wilson. But are there any questions?
I have one comment before a question. And my continual plug to make sure there's no leftovers. Who wants to get some food or drinks while we do the Q&A, you're highly encouraged to do so.
Okay, and the other plug is that if you... Oh, okay, just let me do my plug here. So if you're interested in getting a book, the proceeds go to ODC, and I'm happy to sign anything afterwards. But okay, Peter, you had the first question.
My apologies to this. The answer to this might be obvious to anyone who has a science background, but if I understand PCR, it solves some problem that would otherwise exist if you're trying to compare one DNA sample to another's. PCR it enables a comparison between two samples which otherwise wouldn't be possible. Is that an approximation or no?
Okay, PCR is an enabling technology. You synthesize or amplify millions of copies of something so you can test it. But Peter, you're right in the sense that if the DNA is degraded in two samples, PCR is really the only way that you can amplify the DNA and analyze it.
Okay, so in other words, if you didn't have degraded
Yeah.
DNA, okay, there wouldn't be the problem. What this solves is, and I imagine, in many instances, the DNA that's available for testing has been degraded.
Yeah,
in some way.
well, let me put it this way. If it's degraded, PCR is the only way you can analyze it. If it's not degraded, there are other ways you could analyze it, but I think you'd prefer to analyze it using PCR anyway.
Okay.
Oh, John?
Yeah, Henry, I was struck by the chapter that comments on the hot button issues for
Oh.
databases. Can you make a few comments about that?
Okay, I think I'll try to be brief generally here, but there are different kind of databases. One is a popular, yeah, feel free to eat while we're discussing databases. There are population databases that you need, randomly selected population databases, so that you can interpret a match. So the metric that's used in interpreting a match is called the random match probability. And what that means is, what is the probability that if you picked a random individual from the population, that individual would also match the evidence? So now using DNA, that number can be 1 in 100 million, 1 in a billion. Before DNA, and where you're just doing ABO blood typing, might have been 1 in 10 or 1 in 20. So that's one kind of database. The hot button issues are, if you search a criminal database for a hit, that's fine. But then there's a use called familial searching, where you search a database for a partial hit, and that could be a family member. So some people, the defense bar, has argued that now you're making relatives of the guy in the criminal database a suspect. But my view is you're making them a potential suspect or a person of interest. They're not really a suspect until you build. So getting a match in a database, especially a partial match, is just an investigative lead. It's the beginning of an investigation, not the end. So that's probably what the most controversial aspects are. What kind of databases are appropriate? Now you may have heard of genetic genealogy, where ancestry databases are being searched.
Henry, maybe I'll take one more question.
Okay.
And you made the comment about familiar relationships, and you talked about matching.
Uh-huh.
And what if there were a case where identical twins, neither of which had an alibi, but they're identical? Could
Okay.
PCR be used?
Okay, that's a very interesting question, because I would say until about 10 years ago, it was assumed that genetic testing couldn't distinguish identical twins or monozygotic twins. But now with the new technology called next generation sequencing, if you sequence the genome of two identical monozygotic twins, you can find sequence differences. So that is now possible to distinguish identical twins. Okay, maybe two questions.
Henry, I haven't read the book yet, but I was in your lab when the O.J. Simpson trial was underway. And so my first question is anything about your relationship to the O.J. Simpson trial in the book? Because I know there were some interactions. And if not, why not? If yes, I'm going to start reading tonight.
Well, the answer, Sean, is simple. Yeah, there's a pretty long chapter about the O.J. Simpson case, because I was actually scheduled to testify in the admissibility hearing. So I looked at all the data, and it was pretty overwhelming. I mean, convincing, compelling, whatever adjective you want to use. But as some of you may know, Peter Neufeld and Barry Scheck of the Innocence Project were on the defense side, and they decided to stipulate that DNA technology per se was reliable, so there was no need for a formal admissibility hearing. And they built their defense, as you maybe remember, This was a trial of the century that felt like it lasted for one. They would talk about contamination by the LAPD, or racial bias by the LAPD, or planting evidence. So, as a result, I wasn't called on to testify in that case, but there is a long chapter about it, and a discussion that I had with Peter Neufeld about it. Okay, maybe one last question.
The law is very conservative about accepting new scientific principles, and disability, new scientific principles on the price standard, the other price standard in California. How did you get over that? The law does not appreciate.
No, you're absolutely right. It's a very conservative system, and I think it's the Kelly-Fry standard, which is there has to be a general consensus in the scientific community about something. And I mentioned the case where the FBI wasn't using it yet. I think the judge decided, hey, if the FBI is not using it, maybe there's not a general consensus. But you're absolutely right. It's a very conservative system, and I have kind of mixed feelings about that, because there's probably a lot of cases where new scientific evidence has been excluded that could have been helpful in coming to the correct conclusion.
Well, then we see a lot of allegedly scientific evidence that gets thrown out later, like fire science and things like that.
Yeah, no, actually, it's also a good point that DNA analysis, it's raised the standard and has revealed not only the existence of coerced false confessions, but it's revealed the unreliability of many of the techniques like hair morphology, bite marks, that had been considered to be reliable. Now, I was going to read something about Alan Wilson, but I'm just going to say it in a couple sentences. So Alan Wilson was a pioneer of molecular evolution. He was diagnosed with leukemia, and he went to the Fred Hutchinson Cancer Research Center in Seattle to get a bone marrow transplant. And because Russ and I had developed a technique for genotyping from individual hairs, his family in New Zealand sent hairs from all family members, and I was able to identify one of his brothers as an HLA match. And so Gary flew up from Auckland to Seattle at the Fred Hutch, donated bone marrow, but sadly, Alan Wilson died of an infection. And I just want to conclude this session with saying what a huge impact he had on molecular biology, both in the people from his lab, but also in the scientific legacy he left, and not least of all, the study of ancient DNA, which is one of the most exciting fields, I think, in modern biology. So thanks everybody for your patience, and